So here's a cell. So you guys should already have this on your paper. Here's the cell. So because the cell is considered to be semi-permeable, that means some things can go in while other things can't. So I'm going to pick two shapes. The first shape is going to be a square. This square is allowed to go inside the cell. So this square can move inside the cell, so it's going to show up inside the cell. Let's say I have a different shape. This time I have a triangle. Okay? This triangle wants to go into the cell, but it can't. The cell blocks it. That's because the cell is semi-permeable. So things like water, so H2O, would be kind of like the square. Water can freely go into the cell. This would also include things like salt can go into the cell. Whereas large molecules or things like sugar, they get blocked by the cell membrane. That doesn't mean they can't get in. They just have to find a different way of getting in. So the square can just move in however he wants. Okay? He just slides in. Whereas the triangle gets blocked by the membrane, and we're going to talk about a door in a little bit that has to be open, a type of door that has to be open in the cell to let the triangles through.
I can't see.
Okay, so this time we're looking at another cell. So we have our cell right here, and I want you guys to draw circles all over the cell. These circles represent the heads that we just talked about in the notes. I'm going to take a cross section of just the top part, and we're going to zoom in on it. So this is how you show that you zoom in on something. Okay, when we do this, this is what you can draw when you zoom in on it. Draw the circles going all the way across. Again, what do these circles represent? The heads. And then attached to each head are two tails. So each head has two tails on it. Okay, remember, these heads are what we call hydrophilic, because they love water, whereas these tails are hydrophobic because they hate or they fear water. Okay, now I'm going to do a balloon demonstration to show you what this is actually talking about. Draw that same box from before. I'm going to actually move it to the middle. Okay, and then I'm going to redraw the membrane. So I want you guys doing this in a separate spot so you're not getting all of your words and everything mixed in. Okay, so here's our phospholipid bilayer. This right here, so I'm going to just kind of go all the way across because this is our entire layer. This is outside of the cell. So this would be what down here? If this is outside, this is inside the cell. So outside, inside. Earlier, we used squares. Can the square get through?
Yes. No.
Squares can get through. Okay? Then we had a different one. We had triangles. The triangles got denied, could not get through. But There is a way. Okay? There's something called, guys, there's something called a channel protein. Okay? So I'm going to shade in right here. This is called a channel protein. Channel protein. This time, if the triangle gets through, it is able to pass through the protein and get into the cell. Okay, and that's a process called facilitated diffusion, which we'll talk about on the back side of this note, on these notes.